Namaste to all. This is Dr. Srinath Naganathan, working as Assistant Professor in Biotechnology. So in today's session, we are going to discuss about the sessions which are going to be continued for the molecular biology and rDNA technology. So in this, we are going to see various sessions regarding the DNA, RNA, then protein synthesis. So for DNA, we have to understand that how the genetic material it is and what are the different structures of DNA which have been explained by various scientists. Also, we have to understand the DNA replication process in eukaryotes and prokaryotes. Also, we are going to see about the transcription and translation mechanisms in eukaryotes and prokaryotes, and then we are going to discuss about rDNA technology in detail in our last unit. So, in this session, we are going to see about DNA as the genetic material. So, properties of DNA, if you see, based on the structures, they are being divided as primary, secondary, tertiary, and quaternary. Usually, the primary structure is like a thread of all the nucleotides which have been represented. That is why we say in the primary structure sequences of specific bases like A, G, T, and C. Adenine, guanine, thymine, and cytosine. So the bases are being divided as purines and pyrimidines, which gives us the genetic information. The strand, it actually present as 5' to 3' and 3' to 5' direction. Secondary structure, if you see, it is like a ladder-like structure. When they make a hydrogen bond between the two strands, they form a helic by making a twist. Due to this, they look like a ladder, but in a twisted form. Same way, if they want to make, if the similar nucleotides are present, they can join in between and they can make the last end to form a stem loop structure. So usually the stem loop structure we can observe during our replication as well as during the transcription process when the RNA is being produced. Also, we can also see various structures like pseudoknot in this, where in between they can join and they can form a new structure. Relates to the region of regular conformation of the chain and stabilizes by regular repeating interactions. Tertiary structure, if you see, it is an overall conformation of the chain. That is why normally the DNA are represented as A form, B form, and Z form. During their replication process and during their transcription process, they are being observed in the molecules. Stem loop structure, as I said, it is usually observed during the transcription process. So, in the quaternary structure, they are assembled in a very complex manner. So what happens is we are having like histone proteins like H2A, H2B, H3, and H4. And along with that, over them, they actually make a coiling and they make a supernoid structure. This nucleoid structure, they actually present in the chromosomal model. And RNAs, usually, they have a quaternary structure in a very complex manner, which will be moving here and there in the cytoplasm of prokaryotes as well as in the eukaryotes. So let's see about the backbone of DNA. They are present constant throughout the molecule, consisting of deoxyribonucleic acids. So they are the ribose which makes them for the backbone, and they are linked by the phosphodiester bridge. So in this, what happens is the 3' hydroxyl group which is there, that is the 3' hydroxyl group, it gets paired or adjacent to the moiety of the sugar of one of the deoxyribose group at the 5 prime position, and thereby this forms the 5 3 prime to 5 prime hydroxyl. And in this, the bond formation which is taking place for this is actually the phosphodiester bond. Short nucleotides, we call them as oligonucleotides, and long nucleic acids, we call them as polynucleotides. So base composition of the DNA, if you see, it has been given in 1950 by a team of scientists headed by Erwin Chargaff. That is why this is called as Chargaff's rule. The important conclusion, what they have presented in the Chargaff rule is the sum of the purines and the sum of the pyrimidines are equal. So the purine by pyrimidine is equal to 1. So the adenine plus guanine equal to thymine plus cytosine. And in some cases, it can be methyl cytosine during their replication or during their transcription process. So the ratio of the adenine to thymine is equal to 1. So A by T equal to 1, and guanine to cytosine is 1. So G by C equal to 1. Bases with 6 amino groups are equal to bases with 6 keto groups. That is, A plus C or MC, that is methyl cytosine, is equal to G plus T. So the ratio of A plus T by G plus C is called disymmetric ratio. So based on this disymmetric ratio, we can say varies greatly from one DNA to another. When disymmetric ratio of DNA exceeds 1, the DNA is called AT type. And when the disymmetric ratio of the DNA is less than 1, then it is called GC type. So the total number of the possible nucleotide sequences is 4 to the power of 140 into 10 to the power of 6. Double helical structure of the DNA. Usually, we talk about the double helical structure, that is bDNA, and it was proposed by Watson and Crick in 1953. Along with Watson and Crick, there were one more scientist, that is James D. Watson, Francis Crick, and Maurice Wilkinson. They got the Nobel Prize in Physiology or Medicine in 1962. And this model is actually called as bDNA. So the salient features of the bDNA are the DNA molecule consists of 2 helical polynucleotide chains which are coiled around a common axis in the form of right-handed double helix. That means there are 2 ladder-like structures. You can say consider the ladder. Okay, so this is a ladder. In this, it is twisted. So what happened is it is forming a helical structure, and they are having an axis over which it is in the right hand direction. So the 2 helix are wound in such a way so as to produce 2 interchange spacing of grooves. So what happens is, consider once this is being rotated, they form a groove. That is, the groove is like this. Inside the structure, you can find from a one direction, that is a 2D directional. If you see, it is like groove formation which is taking place. So the wide groove is actually having a width of 12 Armstrong and 8.5 Armstrongs of depth. And the minor groove is a narrow groove with a width of 6 Armstrong and a depth of 7.5 Armstrong. The 2 groove arises because the glycosylic bonds of a base pair are not diametrically opposite each other. The minor groove consists of pyrimidine oxygen 2 and the purines nitrogen 3 of the base pair, and the major groove is on the opposite side of the pair. The major groove is more accessible to proteins interacting with specific sequences of the DNA during their replication and during their transcription process. The 2 helical structures, they wind along the molecule parallel to the phosphodiester backbone. So what is happening is such that the double helical structures cannot be pulled apart and can be separated only by unwinding process during their replication by or transcription by some enzymatic action. That is why they are called as pectonic units. The 2 chain run in the opposite direction. As I said, One, it is 3' to 5' direction. Other, it is 5' to 3' direction. So 3' to 5' and 3' to 5'. So it is like an opposite anti-parallel direction which is taking place, which means that one strand, the ring of the oxygen molecules of the deoxyribose is on the downward. In other one, it is facing upwards. So the phosphate and the deoxyribose units are found on the periphery of the helix, where the purines and pyrimidines bases occur in the center. That means they are actually joined in between. So in this, the plane of the bases is always perpendicular to the helical axis. Diameter of the helix, if you see, it is 20 Armstrong. That is why the distance between the two bases is 3.4 Armstrong in the helix, and between 10 molecules, it is 34 Armstrong. So total, in a turn, you can say it contains approximately 10 nucleotide residue. But if we calculate it, it is 10.5 bases which are present. That means one half base is actually going to the next one. That is why we usually call approximate as 10 nucleotide residue. The two chains are held together by hydrogen bond pairing. So adenine pairs with thymine by 2 hydrogen bonds, and guanine with cytosine by 3 hydrogen bonds. This is called complementarity. The individual hydrogen bonds are weak in nature. So what happens is a large number of them are involved in the confer stability to it. And since the hydrogen bond is a weak bond, sometimes due to the unwinding process during the replication and transcription, it is easy for the enzymes to break open and go for the unwinding process. So if you see the structures as we have discussed, it is actually having the 5' to 3' direction and 3' to 5' direction. So the diameter of this is 20 Armstrong, and this is your major groove, and this is your minor groove. So in this, it is actually having like 3.4 Armstrongs between two bases, and nearly 10 bases are there. So that is almost like 34 Armstrongs which are present. So in a turn, almost like in a whole thing, if we are having 10 bases, approximately we are saying 10 bases, but it is 10.5 5 bases. So the base pairing, as we have said, it is taking place between the bases. So the bases are here the thiamine. So thiamine's oxygen is pairing with the hydrogen which is present in the N3 position. So this is what is actually happening in the adenine to thymine. So hydrogen bond is formed between this. So the first linkages which is formed here is like this. So the guanine to cytosine is 3 hydrogen bonds, and thymine to adenine, it is 2 hydrogen bonds. So the sequence of bases carries the genetic information. The two complementarity chain do not run in the same direction, but they are anti-parallel. So we call them as 5' to 3' and 3' to 5'. The base complementarity of the polypeptide chain, as we have seen, it occurs between the adenine to thymine and between guanine to cytosine only. This means 6 amino purine will always bond with 6 keto pyrimidines, and 6 keto purines will pair with 6 amino pyrimidines. So the base pairing is due to steric and hydrogen bonding. So what is steric factor? The steric restriction is imposed by the regular helical nature of the sugar phosphate backbone of each polypeptide chain. That means the sugar is there, then we are having our phosphate backbone, and along with that the nucleotides are attached at the first position. So what is happening is it is actually pairing with the another one. So steric factors are always present. And hydrogen bonding, as we have seen, purine to pyrimidine, always it is going to be pleasant. So adenine to guanine and adenine to thymine and guanine to cytosine is going to happen. And it is never going to be adenine to cytosine, because there would be 2 hydrogen atoms. One of the bonding position and none at the other. So what happened? Only two will be paired and one will be left open. So there will be unbalance. So imbalance will be always there. So this is what as actually we have explained, that two hydrogen bonds are present always and three hydrogen, and the backbone is always going to be present like this. So phosphate is in the 5' and the 3'. So it is always attached like a zigzag pattern. And due to this pattern, what happens is the backbone, it is looking like not like a straight one, it is like little twisted because the bases are actually going to pair on the opposite direction. So that is why they are always perpendicular to the central axis. So the different structures of the DNA. So the first structure what we are going to see apart from the bDNA is our zDNA. So occurs in the alternating purines and pyrimidine tracts, favors in high salts. That is usually this occurs during the replication process, because at that time the enzymes acts and they start to unwind themselves. So at that time they can form the zDNA conformation. So helps eliminate the electrostatic repulsion of the phosphate group, that is 8 versus 12 Armstrong distance. Methylation of deoxycytidine helps in the formation of the zDNA. Then phosphate backbone forms a zigzag conformation. So zigzag means almost it is going to be, so it is going to be in the zigzag pattern. It is not going to be a helical pattern. So it is like a zigzag pattern which is always present. So the biological importance, it is zDNA conformation, zDNA formation, that is during the transcription of the genes at the transcription sites near the promoters of actively transcribed genes. So it is maximally maximum, it is observed in the eukaryotes during their transcription process. So in the nuclear, there is a nuclear membrane. Inside that, the nucleoplasm is there, where it is going to occur like this. So the negative supercoiling upstream favors the zDNA, and function is to observe the negative supercoiling. And at the end of the transcription, the topoisomerase relaxes the DNA to form back as our helical DNA. So once the transcription is done, it is going to again form as our bDNA. So Z to B conformation. So the zDNA antibodies found in lupus erythematosus and other immune diseases, and double-stranded RNA can adopt the Z conformation. So usually when any virus is entering and it is actually having the double-stranded RNA, sometimes they can have the zDNA conformation. So due to that, sometimes they can again go back to the bDNA conformation and can attach to the normal DNA. So that is why what happens is certain viruses we cannot detect based on this. So the differences between the different forms of DNA, another form of DNA is our aDNA. So I am just giving you a brief idea what is all about. So the helical sense, it is means in what direction it is actually twisting. So basically the bDNA and aDNA, it is twisted in the right-handed direction, but in the zDNA, it is left-handed conformation or direction in which it is rotating. And the diameter, if you see in bDNA, it is 20 Armstrong, as we know. In aDNA, it is 26 Armstrong, and zDNA, it is almost 18 Armstrongs. And base per turn, because in one turn, that is a base per turn means between that how many bases are there. So approximately, as I said, it is 10, but the actual numbers are 10.5 in bDNA, and in A it is 11 and Z it is 12. So the helical rise per base pair, that is 3.4 Armstrongs, that is between two bases, it is 3.4 Armstrongs in bDNA. A it is 2.6 Armstrongs and Z it is 3.7 Armstrongs. So base tilt, that is normal to the helical axis, if you see, it is 6 Armstrongs, but in A it is 20 Armstrongs and Z it is 7 Armstrongs. And sugar plucker conformation, that is in which form it is actually moving, that is the structure formation we will see in the next slide. So in B it is C2' endo, in A it is 3' C3' endo, And Z, it is having both. That is, purines will have C3 prime and pyrimidines will have C2 prime endo formation. So glycoxyl bond formation, conformation, that is anti-parallels. They are anti-parallels. They are having both. That is anti, that is pyrimidines, syn, it is purines. So this is our endo conformation. So in this endo conformation, what happens is, if you see in the A conformation, as we have seen, it is 3 prime. So 3 prime will be uplifted. That is, it is having little up. So what happens is, since 3 prime is little up, the phosphate molecule which is going to bind, it is going to be having little away, little bit. It is going to be away. So the, if you see the Armstrong distance between them, the tilting was almost like 5.9 Armstrong tilt is there. So what happens is, the next base, if they want to bind, it will take little longer. So that is what is actually the endo conformation is. So in this, what happens is, since the 2 prime is little up, so 3 prime is easy now. So 7 Armstrong. So here what happens is, easily the phosphodiester bond formation can take place and the backbone can be maintained. But here, if you see the backbone formation, it is actually going little away. So that is why the distance is actually very high in this. So this is what is actually observed in the ball and stick model. So what I will suggest you is, please refer the different molecular biology books which are available, like Bruce Albert's molecular biology. Then we are having Karp, Gerald Karp's, and various other test books are available, which gives us a brief idea and gives us more details, which you can have for your references. I hope this was interesting and it helped you a lot for understanding. Thank you for giving me and listening to me patiently. Thank you.